talking about Ibrutinib, also known by its brand name Imbruvica. This drug is an anti-neoplastic agent that inhibits the Bruton's tyrosine kinase enzyme, which I'll refer to as BTK. This drug was created by scientists at Celera Genomics as a compound to study the function of the BTK enzyme, and this drug is used to treat various different types of blood cancer, such as chronic lymphotic leukemia and lymphotic lymphoma. So on this slide, I just have the structure of the molecule, and over here we can see these two benzene groups that are linked by an ether linkage. And these benzene groups could interact with other aromatic substituents using pi-pi interactions. The oxygen here could form hydrogen bonds with other hydrogen atoms. If we move up here to the acrylamide group, this is actually the group that's going to bind with our target. Down here we have a 6-membered ring with a nitrogen involved, and this is similar to piperidine, but the difference here is that the nitrogen is binded to a ketone rather than the hydrogen group. The 3D structure just gives us a better look at what the molecule is oriented like in space. So like I said earlier, Ibrutinib binds to an enzyme called Bruton's tyrosine kinase. This enzyme is a member of the SRC-related BTK family of cytoplasmic tyrosine kinases and is required for B-cell receptor signaling, which I'll refer to as BCR. BCR is a B-cell receptor that sticks over the surface of the B-cell in the shape of a Y, and this receptor activates our B-cells by signaling the nucleus, but first it has to travel through the BTK enzyme. This receptor is really important in the growth of B-cells, and when it's overexpressed, it can result in B-cell malignancies, and this is what causes things like cancer. So here we're going to look at the mechanism of action. You can see the BCR sticking over the B-cell here in the shape of a Y. This is where the signal would start, and it would go through to the BTK and then into our nucleus. But here, when the drug is added, it irreversibly inhibits the activity of the enzyme. So by inhibiting the activity of the enzyme here, B-cell activation and signaling are also inhibited. And when this is blocked, the cells stop growing and dividing, which ultimately results in the reduction of the spread of CLL, chronic lymphotic leukemia. And this is why this drug works to treat cancer. So this diagram here shows the interaction between the drug and the BTK enzyme, and here you can see that there's an ATP binding site in the pocket of the enzyme, and inside this pocket is the cysteine 481 residue. When the drug enters the pocket, it covalently binds to the residue, resulting in irreversible inhibition. And then this slide here is just to show you which part of the drug binds to which part of cysteine. So the acrylamide group up here is what will bind with the sulfur of the cysteine molecule, resulting in inhibition.